All right, today we are talking about one of the most revolutionary scientific discoveries of our lifetime. It's called CRISPR. And this isn't just some abstract thing happening in a lab. It's a technology that is fundamentally changing medicine and maybe even our future. So let's jump right in.
And I want to start right there with that headline, because this is not science fiction. In late 2023, it actually happened. For the very first time, regulators like the FDA gave the green light to a therapy that, for all intents and purposes, cures people born with a painful, debilitating genetic disease. I mean, this is a moment that is absolutely going in the history books.
So the big question is, how? How did they pull this off? Well, it wasn't with some giant, complicated machine. It was with a tiny, microscopic tool that was discovered in, of all places, bacteria. Yeah, it's basically a natural defense system that we've figured out how to borrow for our own purposes.
Okay, so let's get into the nitty-gritty of how this actually works. We're going to break down the science of this bacterial immune system we've now turned into a tool that can literally rewrite the code of life. And I promise you, it's way more intuitive than you might think.
So at its core, the most well-known CRISPR system, it's called CRISPR-Cas9. It really is just like a pair of programmable DNA scissors. Just imagine for a second, you have a tool that you could program to find one single misspelled word in an entire library full of books, and then just snip it out with surgical precision. Well, that's exactly what CRISPR-Cas9 does inside our DNA.
And the whole process, as you can see here, is, well, it's pretty elegant. It's basically a two-part system. You have something called a guide RNA. Think of that like a biological GPS. It zeroes in on the exact spot in our 3 billion-letter genetic code that we want to change. Then it brings along the Cas9 protein. Those are the molecular scissors, which makes the cut. It's a super precise search and cut mission.
And you know, this is where the real genius of the discovery comes in. In nature, bacteria were using this as an immune system. They'd grab a snippet of DNA from an invading virus to create a molecular most wanted poster. So if that virus ever showed up again, bam, CRISPR would find it and destroy it. And what scientists like Jennifer Doudna and Emmanuel Charpentier realized was, hey, we can hijack this system and give it any most wanted poster we want. And just like that, a bacterial defense system became a universal tool to edit the DNA of pretty much any living thing.
So that's the science. Now let's talk about the payoff. How is this actually translating into curing diseases that we once thought were completely incurable? This is where the theory smashes into reality and starts changing people's lives.
And this brings us to Casgevy. This is that first ever approved CRISPR therapy I mentioned. It's for sickle cell disease, and the way it works is incredible. Doctors take a patient's own blood stem cells out of their body, use CRISPR to make a tiny edit that tells those cells to start making a healthy form of hemoglobin, and then they infuse those corrected cells right back into the patient.
Okay, so the big question, did it work? Get this, the results were just staggering. In the clinical trials, 90% of sickle cell patients became completely free of the excruciating pain crises that had defined their entire lives. For a disease that had so few options, it's just a total game changer.
Now, taking cells out of the body to edit them is one thing, but the next great frontier is editing genes inside the body. And this chart, this shows the mind-blowing results from a therapy called NTLA-2001. After just a single IV drip, the toxic protein that causes a disease called ATTR amyloidosis dropped by almost 90%. This was the first proof that we could systematically edit genes throughout a living person's body, a huge milestone.
And the progress just keeps coming. In another trial, appropriately named Brilliance, scientists used CRISPR to treat a form of inherited blindness. They injected the therapy directly into patients' eyes to fix the broken gene. And the result? 11 out of 14 patients had meaningful improvements in their vision. They were literally editing genes in the retina to help people see again.
And then there's this story. It's the incredible case of a baby known only as KJ, who was born with a fatal metabolic disorder. In a complete race against the clock, researchers designed a custom, one-of-a-kind CRISPR therapy just for her, targeting a single mutation in her liver. I mean, it is such a powerful look at the ultimate promise here, creating personalized cures even for the rarest of diseases.
But here's the thing. Those original scissors were really just the beginning. The technology is evolving at an absolutely breathtaking pace. Scientists are building out a whole new toolkit of CRISPR editors, and each new version is more precise, more versatile, and capable of doing so much more than just making a simple cut.
This table really shows that evolution beautifully. So we started with Cas9, the scissors,
great for just breaking a gene to shut it off. But then came base editors. These are more like a pencil and an eraser. They can go in and chemically change one single letter of DNA to another without making a big cut. And now we have prime editing, which is basically a true DNA word processor. It can find a genetic sequence, delete it, and rewrite a whole new corrected phrase in its place with incredible precision.
Of course, a technology this powerful, it comes with some serious risks, and it forces us to ask some profound ethical questions. The potential here is absolutely immense, but the stakes couldn't be higher, and we have to be incredibly careful. We're now confronting not just what is scientifically possible, but what is morally and ethically right.
And here's a very stark reminder of those stakes. Remember that NTLA 2001 trial? The one with the amazing results? Well, very recently, that trial was put on a clinical hold. A patient suffered a severe, tragic side effect. It's just a brutal illustration of the razor's edge these researchers are walking, where incredible success and serious, heartbreaking setbacks can happen in the very same study.
So the safety concerns are really real and they're complex. Scientists are worried about things like off-target edits. That's the scissors cutting in the wrong place in the genome. They're also worried about causing unintended damage even at the right spot, or our bodies having an immune reaction to the CRISPR tool itself. And that's not even getting into the challenge of safely delivering it to our cells. Making this safe for everyone is the #1 challenge, period.
And beyond safety, there is a massive ethical line in the sand we all need to understand. Everything we've talked about so far is what's called somatic editing. You can think of it as personalized medicine. It fixes genes in one person's body, but those changes aren't passed on to their children. They die with the patient. But then there's germline editing. This is changing the DNA of an embryo, sperm, or egg, and these changes are permanent. They become part of the human blueprint, passed down through every future generation, forever.
And that line, well, it's already been crossed. In 2018, the entire world was stunned when a scientist named He Jiankui announced he'd created the first gene-edited babies. The backlash from the scientific community was immediate and overwhelming. He was condemned and eventually imprisoned. But now, reports are emerging that he's out and back doing research, which has completely reignited this incredibly difficult debate over where we, as a species, must draw the line.
And I think this quote from Jennifer Doudna, one of the pioneers of CRISPR, just says it all. The power to control our species' genetic future is awesome and terrifying. That dual nature, that's the heart of it. We've unlocked something incredible, and now we face the enormous challenge of figuring out how to handle it.
You know, the scientific question of can we do this has, for the most part, been answered. Yes, we can. The much, much harder question we all now face is, should we? We've essentially been handed the pen to edit our own instruction manual, and now comes the profound responsibility of deciding what story we're going to write.